Hi guys, my name is Sadie Northway-Smith. I'm a senior at Tucson High Magnet School, and I'm going to be telling you all about my SARSEF project. So, we as a species have a trash problem. You guys all probably know this, but I'm going to talk today about a specific type of trash, styrofoam, which you guys all also probably know about, but it's really nasty stuff. It takes approximately 500 years to break down on its own, we think, and it's really, really horrible for the environment. We just keep making styrofoam, so we need a way to kind of deal with this buildup that we've created. And a couple years ago, scientists found a way to deal with that. In a pretty unlikely spot, they discovered that these little guys, mealworms, can actually eat styrofoam. These are the larval form of a darkling beetle, and the researchers discovered that their gut biome is actually what allows for them to break down styrofoam. So they have little bacteria in there that breaks down styrofoam entirely into biowaste. There's no plastic left over. But the researchers of that study, they didn't really investigate the real-world potential of their findings. Can we really use mealworms to break down styrofoam? So that is what I did in my study. I worked with two species, Tenebrio molitor, which is mealworms. Those are the worms that were used in the first study by the researchers, and then Xephobus morio, which are soup worms. That's this guy. That have not really been studied. So it was really exciting for me to get to work with those superworms. First, I established colonies of both species to kind of make sure they could both eat styrofoam, and I recorded a rate of consumption for both species, which was exciting. That was entirely new for Xephobus morio. I also had a pretty interesting finding. The styrofoam-only diet actually doesn't appear to inhibit the reproductive cycle of mealworms, so you can't really see in my colony here, but I have beetles in here, which are part of the life cycle of mealworms, and the worms in here are actually third or fourth generation of styrofoam-only diet. So these guys have never eaten anything except for styrofoam in their life, and they're doing just fine, which is very exciting. But after I established those colonies, I isolated the gut bacteria from both species, and I grew it up in solution and added styrofoam. It was really cool. The bacteria was really, really beautiful. There was some that was this gorgeous lime green, and then another small percentage was actually neon orange, which was super cool to me. But from there, after I added the styrofoam and let the bacteria start to eat it, I kind of began to investigate what factors would affect that rate of consumption, things like temperature, types of incubator, concentration of algae broth that it was grown up in, to kind of try and find a solution to styrofoam waste on an industrial scale. I'm not quite there yet, just because the bacteria take so long to eat the styrofoam, but I'm working on it, and the bacteria do eat, which has some pretty cool potential in the field of bioremediation, which is very exciting to me. So thank you guys for listening, thank you to my teacher for his help and support, and thank you to everyone at SARSEF for fostering my love of science.